The Conversational Flow, a KCAS podcast.
Hello, and welcome to the inaugural episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcast, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow will focus more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotty, and I've been in the flow cytometry space for over 15 years. Started out on the Accuray C6 and BD Fax Jazz, and from there did my PhD work utilizing Canto and Fortessa, looking at novel applications for chimeric antigen receptors, and find myself overseeing operations at the Springhouse Innovation Park site for KCAS. Brian?
Thanks, Adam. Yeah, so Brian Weil, I'm the general manager of the Philadelphia facility for KCAS. I've been in the flow cytometry space for over 12 years. The oldest flow cytometer that I can remember was a BD LSR, I think an LSR2. So definitely been a while, and I've been in the CRO industry now for about 10 years at different places with a focus on scaling and globalizing clinical flow cytometry labs. So definitely excited to be here at the KCS and Flowmetric teams to oversee that process.
And Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe, we are both here in Philadelphia, and Jeremy, our producer, is in Missouri, so we truly are all over the map. We're amped to have you listening to the first ever episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we're looking forward to a fun episode today. To kick us off for episode number 1, Brian is going to give an overview of today's podcast topic a bit before we jump into the news and resources section. Brian?
All right. Thanks, Adam. So today we're going to be going over why flow cytometry needs its own podcast. We'll cover some of the history and some of the ongoing changes that we're seeing in the field. But before we get into that, we'll jump into the news. Exciting week, as always, in the biotech space. We'll go through some medical research news and then jump into some investment news as well. So the 3 items I've got for medical research news are FDA guidance on laboratory-developed tests. So this was brand new here at the beginning of October in 2023, and the FDA is trying to put its foot down to some degree on the proliferation of tests that are coming out as laboratory-developed that may not have all the supporting information behind them. So this is very interesting for the clinical trial space because a lot of the work that we do is very cutting edge and may not have as much time to put a full 510K application behind it. So it'll be very interesting to watch this space, first in the genomic space, of course, but also especially in developing areas like flow cytometry to understand how that impacts diagnostic testing and also clinical trial testing as well. Extremely interested to see how that develops.
Yeah, I'd be curious to see too how that impacts, CAP/CLIA versus some of the support for exploratory clinical trials, as well as secondary endpoints, where we land in that.
That's exactly right. And, we've always really focused on GLP and GCLP type work and relied on that in terms of the World Health Organization guidelines. So these new guidelines, it's always interesting to see how they intertwine and then sort of shape the future of the field. So definitely a recurring theme on this podcast moving forward, I think.
Absolutely.
The second story I've got is just right down the road from us. So two of the recent Nobel laureates in medicine were named at the beginning of October as well. So Drew Weissman and Katalin Kariko were recognized for their work on messenger RNA. And this is hugely impactful for the field, their contribution. So, of course, it was brought to the extreme spotlight with the mRNA vaccines that were developed for COVID. But mRNA has applications pretty much everywhere else in medicine, too. It's really enabling a lot of the CRISPR work to modify genomes for gene therapy. And it's being used for CAR therapy as well. So all the cell therapies that sort of are tangential to the gene therapies, a lot of them are using messenger RNA as a potentially safer route for modification than viruses.
Right. And so there we're looking at half-life, right? Where it may not be, it's not permanent, essentially. mRNA goes in, you get the signal, and then eventually it's stopped. Whereas a lot of the viruses or whatnot, these are things that will persist for much longer periods of time. And so that's one of the allures of the use here, is that you can prime the immune system. That's where these vaccines come from, but it doesn't stay permanent. As far as the expression of the genes from the mRNA.
That's exactly right. So definitely a very interesting use case. Usually you want drugs to stick around for a long time, but here with viruses, they're just too good at their job, right? So we need something that is a little bit less permanent and a little bit more controllable during the course of the trial. So that's what these two have really brought to the forefront and are continuing to develop in their labs and in their research. So looking forward to what comes next. Also, of course, a shout out to their Philadelphia location. Just speaks to the incredible talent we've got in the area. So always want to recognize the hometown team there in terms of research contributions. Last up on medical research news, I just wanted to highlight that it is indeed conference season. So, of course, the KCAS team will be at a number of conferences like Meeting on the Mesa, AAPS, MetroFlow. But our work through our sponsors is also getting published at other conferences like ESMO and Festival of Biologics and others. So I'm extremely excited to see all the new developments that come from both industry and academia at these conferences and understand what the field is really looking towards for the next set of challenges.
Yeah, it's some exciting travel as well. So we've already been to Cyto this year up in Montreal. Our team was there, had a great time, made a lot of good connections, learned new things. We've got AAPS, I believe that's down in Florida, right?
That's right.
Meeting on the Mesa in California. And then closest to our heart would be MetroFlow, which is much more local for us, New York area.
Yeah. So a lot of good things, and there's no replacement for the conversations you can have in person at all these conferences. So really looking forward to meeting everyone and seeing everybody back at these major events. Switching on over to investments, a couple things that we're tracking. One on the vendor side. So here we, of course, at KCAS have an isoplexis instrument. That company went through a number of different transformations as it was sort of merged with Berkeley Lights into the Phenome X company, and now the entire conglomerate has been purchased by Bruker. And I highlight this as an interesting step because there's been a lot of development in the single cell analysis technologies. So isoplexis is, of course, a great example of one. And of course, flow cytometry is probably the oldest of these techniques, but there are a number of new techniques that we're keeping our eyes on, like the 10x systems and mission bio systems that are really bringing the promise of omics towards single cell analysis. And it'll be interesting to see both how this shapes up from an investment perspective and how this becomes more robust and scalable and potentially applicable to more clinical trial testing.
Yeah, and we actually, speaking of Cyto, had presented a poster with the isoplexis technology up at Cyto in Montreal back in May. The interest there is the ability to probe not just intracellularly, the supernatant as well, and get a good idea on cytokine expression to really understand the drivers there, therapeutics, and how they're impacting the body, the extracellular space and intracellular space.
That's right. And there's no other technology that can do what they do. So also the field's struggling then. How do you then interpret that data and then apply it towards your research questions? And I think that's the next frontier that Flow is also now addressing with some of the AI technologies that they're bringing into spectral analysis. So definitely an interesting space to watch and something we'll keep a close eye on. Also near and dear to our heart, of course, is the BMS purchase of Mirati Therapeutics. So that was a pretty significant purchase, somewhere north of 5.7 billion, if I remember correctly, and shows that even though the biotech space is still, to a large degree, in a downturn, when there's a strong therapeutic target, it can definitely command a significant price. Definitely, BMS being a premier provider of oncology drug development and drugs in general, it makes a lot of sense for them to bring in a company like Mirati that can broaden their pipeline and also give them some additional capabilities there. So we'll definitely be looking with great interest in terms of how that helps BMS position themselves commercially and what that means for the future of commercial clinical trials with regards to how we target therapeutics and what is considered a really strong commercially viable therapeutic as well.
Yeah, and this is in the area of non-small cell lung cancer, targeting KRAS, specific mutation, and has been historically something that was difficult to target. And so looking for the further developments, and it's been improved, back in 2022, I think it was, December of 2022. And so, yeah, bringing to bear the resources of BMS behind certainly will be a good thing.
Yeah, I mean, KRAS can have its own podcast by itself, so looking forward to that one as well.
And then shifting over, so KCAS News, we're starting a new podcast. Brian and I, we are at the SHIP site, so the Springhouse Innovation Park. We've been here now, the Flowmetric team, since the end of 2022. And are now wanted to get the message out, reach a broader audience, let everybody know what we do, and interact with clients, friends, interested parties, scientists, flow cytometrists for sure, is something that's really excited about.
Yeah. I'm particularly excited about it because it's not a TikTok. My dancing skills are not up to snuff, and I don't know if I can really do interpretive dance to demonstrate spectral flow, Adam. I don't know about yourself.
It's funny you mentioned that. As I'm driving today, I saw a car in front of me that has a bumper sticker, "Caution, driver singing," and I was thinking, so glad they didn't ask us to sing. I can talk, but nobody wants to hear me sing. So completely agree there. As part of this, so we've already got some news, updates on investments. We'll have specific news to KCAS Flowmetric. We'd also like to bring some scientific curiosity, right? And so I'd like to introduce a paper of the month section. This month, we've identified Flow Cytometry, the next revolution. This is a review paper that's in the journal Cells. That journal has an impact factor of 7.666. Very, very specific there, the 666 there. But interesting, it just came out. It was published this quarter, 2023, and goes over the history of flow cytometry, very much looking at the different applications for it and where it's going in the future. It's obviously very important to us because it's focusing on flow cytometry. But one of the other things that they point out is the direction of spectral flow cytometry and how that is enhancing the role of what we do in the industry by allowing us to interrogate a much higher number of parameters. And so, Brian, what did you think about this paper?
What I love most about this paper is that every 5 or 10 years, there will be a really solid review paper that covers everything in great depth. And every 5 to 10 years, our perspective on what mattered at that time changes completely. So if you had written this 5 or 10 years ago, you would not be focusing on spectral flow because it was such a small portion of the market back then. But at this point, it's becoming a greater and greater concern, and always interesting to see how you can, now that we know that it's important, then track back exactly how it was growing. Much like the mRNA story, right, with the Nobel Prize. At first, it was a research backwater, and now it's huge. So by the same token, definitely seeing how the review treats all the current developments is very interesting, and I thought it was very well written.
Yeah, I certainly would agree. It's out of Purdue University, well known for their expertise in flow cytometry over there. And so certainly appreciate the perspectives that they're bringing here. Many of the applications we do on a day-to-day basis here at Flowmetric KCAS. And so it's exciting to see them there. It also walks through parallels of development in drug discovery, for example, going from looking at was it antibiotic therapy to monoclonal antibody therapy to next steps in bispecifics, and now what we're seeing a shift in treatments towards personalized therapies and cell therapies such as chimeric antigen receptors, and just mirroring how technology just continues to drive us forward in health and therapies there.
Sure. And, in our daily work, we often get pigeonholed into exactly what we're working on and why we're working on it. And it can be just nice and refreshing and sort of reinvigorate your scientific curiosity to learn about other paths and how they're using the same tool for a different purpose. But also potentially a really useful thought of, hey, I've never thought about using flow cytometry for this purpose, but I see those crazy antibody people are doing it for this. Maybe I can do the same thing with my therapy. So always a useful read there.
Yeah, and the other thing that it brings to bear is the AI as part of the next step, right? The issues that we're seeing right now are the increase in the amount of data that is being generated, and how do you analyze all of that? And so more and more you're seeing machine learning come to the forefront. And obviously, Brian, we've had some fun with this going into ChatGPT, for example, and it's amazing what it can spit out just from simple questions, and then applying something like that, not specifically that itself, towards analysis. And what it does is it learns. Now, flow cytometry certainly is unique in and of the fact that there are many ways to analyze data. You talk to different groups and they handle things differently. And so that's going to be a challenge as we generate machine learning. But more and more you're seeing that acceptance because it's necessary, right? At some point, the time required to analyze every single large data set would just be monstrous.
And that's the second time we've mentioned AI on this podcast so far. So what is that, 10 minutes, give or take? So I'm sensing yet another podcast. And, basically everything that you were just discussing, there's 100 different points to talk about with how can we trust these algorithms? How can we build them in a reliable way? How can we apply them to a clinical trial? And then defend their use to the FDA and make sure that they're doing what they're meant to do, right? Like enable faster and better decision making. Mm-hmm. And definitely a huge issue and a huge focus of the field right now. So looking forward to talking more on it. But before we go into that, of course, why don't we jump over maybe to the main portion of our podcast? And Adam, if you want to kick it off there.
Yeah, why does flow cytometry need its own podcast? Well, come on, we're talking about lasers, right? And cells, immune system. It's just, why wouldn't you have a podcast around flow cytometry? I've been involved with flow cytometry for quite some time. It's something that just is such a powerful tool and continues to own broader applications. And so, yeah, Brian, why do you think, why flow cytometry?
Yeah, I mean, it is such a massive part of clinical trials now, and it's becoming ever larger. Knowing how the immune system interacts with new drugs, knowing how the immune system can be either a prognosticator or an important factor to review in terms of who the responders were and why. So really teasing out the mechanism of action of what's going on during a clinical trial is essential. So having a space to discuss this at length Yes, we can write papers, we can write blogs about very focused topics that'll be useful to the community. But I think a discussion and a place for people to come and bring their questions and then we can give feedback to is essential for the field. So definitely looking forward to hearing more from the community about what they need and what they need to hear.
Yeah, feedback. It's very important for. It's one of the lifebloods of a podcast, and would love to hear from our audience. Imagine right now the audience is going to be a bit smaller than hopefully it will be in the future, but would love to have questions that we can bring here and answer as best we can, or identify resources that can get us the answers. And so going from there, why is flow more prominent now than it had been in the past?
It is a mixture of things, right? It is the technology that we have now enables us to do so much more. So that's definitely a significant factor that we, of course, follow in the lab. But also, I'd say the recognition of the importance of it is significantly improved as well. So, seeing how the immune system was critical to developing a lot of drugs then made other people realize, hey, I need to include flow cytometry in my trial. And the proliferation of people that understand flow and then have gone to new companies. We've really seen that through many of our clients, right? Where one of our clients will start out at one company, jump to another company, realize that they need to start up a flow program to understand their drug's mechanism of action, work with us there, and then jump to yet another company. And it's that realization, I think, amongst the pharma community about how important flow cytometry based endpoints can be and how powerful they can be in developing not only the current drug that you're working on, but understanding the mechanism of action to develop all future generations of drugs. That's really driven the increased prominence.
Yeah, and you touched on a point, so true. A community, right? Not just pharma, flow cytometry. When you're going to conferences, we're running into a lot of the same people. We've got clients coming back, whether they had gone to a different company or a different group in their own company. It's a community, and love to see the returners. Also love new faces. And so with that, you learn a lot too on both sides there. One of the most rewarding things I have to say, though, is when we get feedback from clients that some of the data that we provided aided them in furthering development of a therapeutic and understanding mechanisms of action, or really it's why you get into science, right? And especially why you get into the CRO business, pharmaceutical, biopharma, is to help people. And so when you get that, it really hits home.
Yeah, and towards your point about just community and prominence in general, that whole cycle is really making both sides of the coin significantly better, right? So the client side is asking for more and more standardization and rigor, but at the same time also complexity. And then the CRO side is advertising out this and driving standardization and labor improvements to make things much more robust and automated and reproducible. So with both sides really pushing on their strong points, I think that is allowing it to be used in more cases and allowing it to leverage all the new technologies as it comes in. That way, at least we know what questions ask because we've been down this path before.
Yeah, what you're highlighting is this whole thing is a partnership. We're working together to meet a goal. It's not about throwing things over the wall. It's not about just trying to rush through things. Everybody's got timelines, right? And we certainly observe timelines, do our best to meet and even exceed there. But at the same time, making sure that there's communication there, and all centered around flow cytometry. Really brings me back to when I started out with flow cytometry. The first instrument I was on was a BD Fax Jazz. And for anybody who's not familiar, that's a sorter that is anything but automated. Everything is a button or a knob. You've got to do your own AccuDrop and align lasers. I remember getting that machine in. It was one of the most intimidating things because before that, I was really only running on an Accuray C6 and went out to BD in Milpitas and did the training. And that really was where I fell in love with flow cytometry. How did you get into it, Brian?
Yeah, also on a BD, an Accuray C6. I think that was before BD purchased Accuray, actually.
Mm-hmm.
And yeah, just a compact little instrument that could spit out so much data on all of my research questions. Anything from just a simple how effective was my transfection to much more complicated. How well did these stem cells differentiate in response to this condition? And how does that compare to all the other differentiation conditions? So the fact that I was able to answer all the questions so quickly and with so much statistical data and backing, definitely made me fall in love with it, as you say. And then I started getting into the larger and larger instruments and realizing what additional things that they could do and how quickly they could map out really complex cell subsets.
So what you're saying is the C6 is basically the same thing as the Cytek Aurora, right?
I mean, they both start with C, so they have to be, right?
Right.
No, so I'd say the C6 is a gateway drug to flow cytometry. Right?
It is,
so,
yeah.
The C6 and maybe like a MyFlow or something like that would be, these sort of smaller, less complex instruments. And then you just build from there, and then you get into, let's say, the 10 color range. You realize you can do more than that. You can get into the 20, and then all of a sudden you go into spectral flow, and just blows your mind on how all of these things can interact, both experimentally with how antibodies can potentially interfere with each other or bind, how their fluorophores can interfere or have spectral overlap, and then how the instruments can deal with all that and have special techniques to filter it out. So definitely it's a steep learning curve, but some of those intro instruments are great for starting someone out there.
Yeah, imagine starting out on a Cytek Aurora. I mean, all the colors there. I think having gone through it from a C6, a Fax Jazz, to a Canto, to a Fortessa, and then to a Cytek, you appreciate each stage. You appreciate the differences in the machine, understanding compensation versus spectral unmixing. And now just diving in, I guess it's like somebody being born and being a teenager, just having a cell phone in your hand from day one, right?
Right. An iPhone, not even like the old flip phones that we used to have with the 1.2 megapixel cameras or whatnot. Yeah. So definitely a sea change there.
Yeah, for sure.
And, KCAS has been there since the beginning, of course. We've been more than 11 years now at our Philadelphia site, and KCAS has been in the Kansas City area for 40 years. So we've seen a lot of these developments come and go, and now we're on the forefront of it with the Cytek. So excited to see what's next and how we can do it, whether that's on the software side with AI or with new hardware.
Yeah, it's a great summary there.
So that concludes our main portion. Ordinarily at this step, we would have some feedback from fans and questions. As you can probably imagine, we don't have any fans or questions as of yet. So we will just move right on to the next step.
Brian, this is Jeremy, producer here. People don't usually hear my voice, but they may know me from our other podcast, the Weekly Bioanalysis. Speaking of which, we actually do have a couple of pieces of feedback. The hosts over there, Dom and John, both wanted to say something to you guys. So this is Dominic Warino, says, "Best of luck, Adam and Brian. Have fun and go with the flow."
That's great. Absolutely right with the theme there. Thank you, Dom.
Very Dominic, yes. John Perkins, the co-host over there at the Weekly Bioanalysis, says, "Best of luck with the first episode of your new podcast. Here's to many more. Maybe we can even join for a chat about mass cytometry sometime."
Absolutely. I mean, here we are. We've only been here for like 30 minutes and already people are joining in. So great.
Yes.
Yeah,
And in
Jeremy,
fact,
how many episodes have they gotten so far?
so we just published episode 70 this last month, and a little sizzle for the steak here. I think episode 71 may include a little something special related to this podcast, but we'll get to that later on. I don't want to
Very
steal any
exciting.
thunder.
Mhm.
Last up, we have John Buckseth, CEO of KCAS. This is kind of a long one. Says, "Hi, Brian. Hi, Adam. I just want to congratulate you on all the work that you gentlemen have been putting in to advance the company's capabilities, expertise, and facilities to support our clients in their efforts to bring life-saving drugs to the market. The progress has been made under your leadership, and it is phenomenal. Looking forward to listening to the podcast to hear what's next for KCAS in this crucial technology that is so critical in successfully developing the drugs of our future. Also, congrats on rolling out your new EQMS system. Looking forward to hearing what you have planned next."
Excellent. Well, appreciate all the words of support there. Definitely, we have been extremely busy, both Adam and I, and everyone at the Philadelphia site, in rolling out new equipment, new software, new processes. So we are ready and able to scale with our sponsors and really fulfill this growth plan of KCAS. So we're really looking to work with any and all sponsors that need flow cytometry, and we think we are the ideal partnership, and we've built something that will help everyone really achieve their goals, and that's why we're doing this. First of all, building the infrastructure itself, but then also building the podcast to help people understand that yes, we can absolutely help you, and if you have something that can be done with a flow cytometer, we are the group to do that for you. So looking forward to building that out and seeing how that develops.
And key to that is it doesn't stop there, right? We're continuing to listen to our clients, listen to the industry so that we can get better, bring more technologies on, apply more rigor, and really drive the state of flow cytometry and support for great science forward.
That's right. And just like our paper of the month, every 5 years we look back and we see different things, right? So if we stay still at any point, we would lose. So absolutely need to continue to be on the cutting edge of things and look for different ways to help and partner with clients to answer these research questions and make sure that we develop the drugs in the right way and fully understand these mechanisms of action.
All right, Jeremy, any other feedback from fans, or should we move on to our next stage?
That's it for now. Feel free to move right along.
All right. We've already gone through paper of the month. We've got some other ideas coming up as far as next papers that we'll be discussing. Remind me, what are we thinking of for next paper?
For next paper, we've got a couple options. I think one of the most relevant, and it sounds like what John Perkins might want the most, is a comparison of mass cytometry to spectral flow cytometry to understand what the differences are, how they can be combined, and what the advantages could be for one versus another. So definitely I'd see a couple of different really good publications comparing and contrasting the two techniques.
But there's no rumor there, the truth to the rumor that Jeremy's going to have like hissing and booing and cheering tracks as we go back and forth between these technologies, right?
I mean, a new development. I think we need a laugh track for this. I think it worked for Friends. It'll work for us.
I love it.
A laugh track. I'm a little terrified now. Excellent. Yeah, so that sounds like a great paper. Looking forward to the discussions there. We can also post that link so that people, if they wanted to get eyes on the paper before we discuss it, have that opportunity, right?
Yeah, absolutely.
Okay. So what do you got plans for the weekend?
I've got a friend coming in to town, so I'll have to show him the sites and things like that. So we usually end up doing some light axe throwing or perhaps beverage drinking of some sort. We'll see.
Beverage drinking. So do you have a specific beverage that you're looking at?
We've got a couple breweries that we usually hit up, and we've been going more and more towards. We were in the sour phase for summer, but now we're going into the IPA phase, I think, for the winter. What about
Oh,
yourself? Big weekend plans or beverage choices?
well, the beverage choice. So one of the courses that I ended up taking during grad school was in a literature review of beer brewing, right? And so one of the things that was really interesting at that point in time was the director of that program over there actually identified what was it, a species of yeast that generates just directly sour beer. As you know, a lot of the beers that are made that are sour generally have some sort of bacterial component to this, but this was from the yeast fermentation. And so just giving a shout out to Dr. Farber there and his work for beer brewing. But yeah, as far as myself, man, I'm all over the map. Lately I've been into spiked teas, but I've also been into Southern Tier, Warlock, just owing to the season. And I will say too, bourbons. So Angel's Envy, as you know, we've been going back and forth. So many people in this area seem to really love it, and found one of their new offerings that really is delicious.
Nice. And see, science working itself into our everyday life. All the time I find myself trying to explain science topics, or in some cases my job, to family. And with a 6-year-old, I've got to keep it pretty high level. And I usually try to tell them that I help doctors understand why people are sick. So that's the one sentence definition of what I think we do and how I describe it to my family. How do you talk to your folks about that?
So, as you know, I've worn a lot of hats over my career. Most of it has been in the lab, the vast majority of it, but I've also done project management. And I would say project management was a lot more difficult for me to describe to my family. In the end, I do think that I was able to get across to them that I help guide projects and, understand the importance of project management there. As far as my current job, I find it a bit easier because everybody knows scientist, right? When you say scientist, it conjures a picture. Now, in my current state and age, maybe nearing more of like the mad scientist look. And again, with the Halloween thing, maybe just get the gray hair and the lab coat. And I really don't need to dress up all that much for that role.
See the benefits. You got a built-in Halloween costume. That being said, I did think being a scientist would involve many more multicolored solutions. It was disappointing to learn how many of them are clear.
But hey, I mean, to be fair, that's probably why you ended up going to flow cytometry, because
True.
some of those vials do have some really cool looking colors.
That's right.
Yeah, I liked when I was at a previous role doing transfections and inserting GFP into cells, and you can look in a fluorescent scope, and it's not just GFP, green fluorescent protein, but there are other colors as well, and you could it's beautiful to look at, and it's just another reason why fluorescence and lasers just is so captivating.
Yeah, an art form to it. Absolutely.
Mm-hmm. So we've got next recording looking like we'll be putting something together in November. As Jeremy had mentioned, we'll be having Dom and John join us from the original KCAS podcast. Is that an accurate way to describe it?
I think so. They are the first.
All right. Excellent. And so we'll look forward to that. Dom and John definitely cast a big shadow. So we've got something to live up to there. Excited to have them on, hear what they have to say. We'll go over the paper that we mentioned about mass cytometry versus flow cytometry, comparisons, benefits, why would you go either direction, and really dive into all things flow cytometry there. Anything else, Brian, that you wanted to leave our audience with?
No, of course we'll give some feedback on all the conferences that we're going to and things like that, but really great to get our inaugural episode out there. Hopefully it generates some questions and some new topics for us to bring up. And we'll look forward to talking to everybody else soon.
Excellent. Thank you.